Thank you very much, Shane, for the introduction. So I realize now, having listened to the other two talks, there's probably a lot of stuff I should have included, including my academic journey, which was very unconventional. So very briefly, I'm from Namibia originally, so I came down to study at Stellenbosch University in 1997. So after finishing my undergrad, I did honors in microbiology and did a master's in microbiology. And my professor desperately wanted me to stay on and do a PhD, but for reasons of life, I saw my brother just had matric, and he went and worked in a bank, and he was already buying a car and a house and everything, and I'm like, I need to catch up. So there's a reality to studying that they don't tell you about. And I started working for my then professor for the next 5 years, and he was nearing retirement, and then eventually middle 2008, I moved to University of the Western Cape to start working there as a researcher. So where I joined is called IMBM. You'll see we're wearing the shirts today. So this is the Institute for Microbial Biotechnology and Metagenomics. So the two previous speakers told you about larger things, so macrofauna. So now we're going to turn our focus to smaller things, so microbes. And maybe at this point in your life, you've got some idea, bacteria are around, but I don't think that people, especially young people, quite realize the important role of microbes in everything. So ecology as well as our health and well-being. So the microbiome has become something that's more in the popular culture these days, and I think we all now understand that our gut is really important to our health and well-being. But what I'm going to tell you about today is, so we're not marine ecologists. We're a fundamental research lab, so we just tinker around. We try and understand biology at the fundamental level. And in so doing, we then try and use some of that information to develop products or services. What I'm going to tell you about is just the genome sequencing of a bunch of these bacteria that we isolated from some sea sponges at Kalk Bay. All right, just a very brief introduction to the Institute. So it's headed by Professor Marla Trindad. So you would have seen her around. She's currently in the lab. And then we've got a couple of academic staff members. So we've got Dr. Nompumelelo Nyembe and Rian Den Haan. We've also got Dr. Anita Berger, who's our innovation manager. So if we find something really interesting in the lab working on these bacteria, then it's Anita's job to go out there and find companies who may want to work with us or partner with us to try and commercialize this. I am the lab manager, and as Shane indicated, we also run a flow cytometry and sequencing facility, and I'm the manager of that. And then we've got Chantel, who's an administrator, Stephanie, who is our biobank manager, and you will see her in the lab. She'll help you guys to isolate the DNA. So a couple of years ago, we were awarded one of the biodiversity biobanks of South Africa, and that is because we have a huge collection of microbes that we've isolated from a wide variety of extreme environments. As part of, as Shane explained, the 1K SA project, we committed to sequencing 500 of those bacteria in the collection. And so we've surpassed that number now. And then Carmen is our technical assistant. We actually run a very nice lab for the students. Usually if you go into a postgrad lab, you'd have to pack tips and wash your own glassware and all that good stuff. And Carmen makes sure that none of the students have to worry about that. They can just get on with doing the work that they want or need. All right, so the Institute we're interested in a very wide array of things. I often joke and say that the list of things we do is much longer than the list of things that we don't do or have tried to do. So this slide just tries to capture some of these projects. So at the top left here, we've got some nice images of bacterial viruses. I don't know if you guys have ever seen those. These are called bacteriophages. And there we can see some genome alignments from these guys. So you can break open their head that contains the DNA, and you can sequence that DNA, and then you can make some comparisons to see how similar or different they are, for example. Another project that we were interested in is there's something called American foul brood disease, and it affects honeybees, also here in the Western Cape. So it is a bacterium called Pedibacillus that'll infect the larvae and kill them. So we were actually looking to isolate some viruses that can kill the bacterium that kills the honeybee. So top right here, you can see I'm balding already there. But here we are in Bella Bella, so this is warm baths. And we were trying to isolate bacteria that make these little structures that you see here. So these are actually little bar magnets. You know, like your fridge magnet? The bacteria actually makes these little magnets inside. And they all line up end to end. And the bacteria use them to orient themselves in a water column at the right oxygen tension so they can grow. So this guy over here is a French researcher, one of our collaborators, and what they were looking to do is to turn these into contrast agents for MRIs. So you may not, so I can see there's one excited expression. So these little magnets you can actually functionalize, you can put all sorts of things on them. So very exciting work. So this over here, we were trying to improve the evaporation rate in mining so-called brine ponds by cultivating pigmented bacteria to increase the solar absorption and heat the water slightly to try and increase the evaporation rate because they want to get rid of that water ASAP. Another theme that sort of emerged over the last couple of years in the lab, and this is actually the one that's closest for us to commercialization, is biosurfactants. So do you guys know what a surfactant is? Soaps, sunlight. But you actually get bacteria that make compounds that are amphiphilic. So they've got a fat-loving part and they've got a water-loving part, and that makes them great for any kind of soap. And of course, you will know or appreciate that most of these kind of compounds come from crude oil at the moment. So your petrochemicals, and we're looking to replace those with more natural, renewable sources. All right, so we've also got a big program in the past, at least, looking at plant-microbe interaction. So here we've got some rooibos plants. So Dr. Brian Kirby, who's just recently left the Institute, had a big program looking at the microbes associated with the roots of the rooibos plant and aloe species in particular. All right, okay, so what we're going to talk about today and what you're going to be doing in the lab next door is extracting DNA from microorganisms. So I thought I'd just have a slide to kind of remind us of the structure of DNA, because a little bit later I have got a couple of slides that talks about the nanopore sequencing technology that Shane mentioned. And I want to show you this because it's absolutely mind-blowing. If you're interested in science and you're interested in biotechnology in any degree, this is like the culmination. Like, even for me as a seasoned researcher, the amount of development that had to go into this technology is literally mind-blowing. So I have to show you guys this stuff. So hopefully most of you know the structure of DNA. So we've got this double helix, these two anti-parallel strands, and there's a direction to each one. We kind of read them 5' to 3'. And of course in DNA we've got deoxy, so there's a hydroxyl group missing there, ribose, and we've got these ribose phosphate backbone that runs down the side of the DNA. And then we've got these pyrimidine and purine bases that link up. And we've got base pairs with 2 hydrogen bonds and those with 3. And the structure of this molecule dictates a lot of things that happens inside the cell in terms of how the DNA gets replicated, so on and so forth. But when it comes to the technology I'm going to show you, it's important to know that you've got these 2 strands and we're going to separate them, and one of these is what will move through the pore. I'm going to show you a little video to get sequenced, to determine the sequence of those base pairs. So the other thing that, I tried to put myself back in your shoes. What did I know? And I guess you guys might be smarter than me at this point in life, or when I was your age, but I didn't have a feel for the size of things. So, I mean, you heard about a whale that's like, how many meters long? 27 meters? That's, it's pretty big. And even then, I think we don't have a sense of scale. If you stood next to the whale, you'd start to appreciate it. But when you talk about small things, I think you have even less of a feel. So I always tell our students, the first time you open a biochemistry handbook, the first page kind of tries to show you this is the size of a water molecule, this is the size of a virus, so on and so forth, so that you develop that sense of how big these things are. So over here, top left, I show you a graph that kind of shows you how much DNA different organisms contain. And you can see at the top here, we've got mycoplasma, so it's like a microbe, and it has maybe like 1 million base pairs. So if you now cast your mind back to the previous slide, every pair of bases, so there's one base pair, and so it's got 1 million. And then you can see for gram-positive or gram-negative bacteria, slightly bigger, so that's in the range of, say 5 million base pairs to 12 million base pairs. And then we go all the way up here to flowering plants, which have huge genomes. And of course, we've got the human for comparison, 3.2 giga base pairs. So this is a lot of sequence information. And again, plasmids, so these are little circular pieces of DNA that you can find in bacteria in the range of 5 kb to 150. And the bacteriophages that I mentioned earlier, you can see what size they are. But so what's the deal with DNA? I mean, what makes it so special, and why would you be interested in it? In preparation for this, I asked ChatGPT to give me a nice table that sort of summarizes, some of the things that you can do if you had a piece of DNA sequence in hand. And so it's a fairly extensive table. I've just highlighted some things that we tend to do with DNA in the lab. So here you can see under the sort of field or category of genomic research, we can make so-called plasmid or backed libraries, and we can screen that for metabolic traits or genes of interest. Now I appreciate, when I say something like that, you don't even know what a plasmid library is. But one of the techniques that is developed over the last sort of 20, 25 years is something called metagenomics. It's right there in the name. And I'll explain a little bit later what it is, but in a nutshell, you can now go and grab, say, a gram of soil outside here, and you can extract all the DNA from there. I can then take those pieces of DNA and I can clone them into what we call a vector, and I can put that in friendly E. coli, or friendly bacterium like E. coli, and I can cultivate those pieces of environmental DNA in my lab. And if there's a gene on there that makes an enzyme that I'm interested in, I can screen it in some way, and I can try and find the piece of DNA that gives me like a new enzyme. Very powerful technology. So then, of course, what you've heard about today is environmental monitoring. So in the past, we used to be interested in looking at the diversity of bacteria that we would find in any kind of environment. So there is a gene inside the organisms called the 16S ribosomal RNA gene. And in the past, people would just amplify that one gene and sequence it, and they could get an idea of the diversity. Today, we would probably sequence absolutely everything that's in the sample to get the full picture. And then I thought, quite interesting, right down the bottom here, it says education and citizen science, kits for schools and public projects. So so this too is an application of, if you can sequence some DNA. All right. But again, I thought what I'd do is to zoom in on one aspect of what we can do with DNA. And I think, again, we all appreciate that organisms in hospitals and so on are becoming resistant to the different antibiotics that we want to treat them with. So I'm going to walk you through how we go about trying to find novel antimicrobial compounds from these bacteria that we find in a marine environment. So here I just show you, the classic example of penicillium and how it can get knocked out by other organisms growing around it. So here you can see it covers the plate. As soon as we've got these organisms dotted around, they are producing some sort of compound that inhibits the growth of the fungus. All right. So I thought I'd also just take a little pause and tell you guys about DNA sequencing, and particularly at the University of the Western Cape. So yeah, you can see some of the dates dotted here. I don't know when you guys were born. Quickly give me a spin around the room. 2007. All right, so you can see yeah, so when I joined University of the Western Cape, and this is not really appreciated, that this university has actually been at the forefront of next generation sequencing in South Africa since its inception. So the machine that's pictured over here is called the Solexa Genome Analyzer. This was actually one of the first next generation sequencing machines in the country. And one of the professors at the university at that time wrote a grant, and they managed to buy this. And here I just show the sort of specifications of the machine. So this could output 25 gigabases. So if you cast your mind back to that human genome, it's 3.2 gigabases. This could output 25 gigabase. But the read length, the piece of DNA that we could sequence, was only 75 base pairs long. And so if you cast your mind back again to what Shane said, my puzzle piece is now in all these little 75 base pair fragments. My human genome is 3.2 gigabases, 3.2 billion base pairs. Now I've got to remake this, complete picture from 75 bases. All right, so fast forward several years, and we were successful in buying one of these machines. So this name might be familiar to some of you. Maybe if you look at the medicine cabinet, Roche. So they brought out a technology called 454. And so here you can see now the output is much, much smaller. So this is megabases, this is gigabases, but the read length had improved substantially, 450 base pairs. But because we work with microorganisms, the genome's also much smaller. So we didn't need a machine this size. This worked absolutely fine for what we wanted to do at the time. But for various reasons, we then decided to move on, buy a different technology. And in 2012, we managed to get a so-called Illumina MiSeq. So this company, Illumina, today is the largest. They sell the most sequences, and they've got the biggest so-called install base. But the company predominantly makes short read sequences. So you can see that the read length is about 300 base pairs here. And now we're back up, we can get 15 gigabase per run output from this machine. And again, this served us very well for a very long time. But having those short reads, there's an intrinsic problem with it if you want to try and put your genome back together. In these genomes, in particular plant genomes and high eukaryotes, you get a lot of repetitive regions. And if the read length is too short, we can't span that region, and we can't correctly put the genome back together. And so people went on and developed so-called long read technology. And so today, the one I'm going to show you, and Shane mentioned, was Oxford nanopore technology. This mind-blowing thing that has happened. So now you can see, look at the guy's hand for reference. This is like a little box like this, and that'll give you 500 gigabases. So you won't appreciate the size of this machine. This is basically the size of a room. So the one corner was taken up by the server, and the rest of it was essentially like a wall, this thing. It was a huge machine. And today we can do multiple times of this with a little box this size, and you're going to see an even bigger version of this inside. And now we can sequence a single read. So 300 base pairs, now we're up to 4 million base pairs for a single read. Again, if you guys don't necessarily have the context to understand how magical this is, but it is mind-blowing stuff. Right. Okay, and you have to be proud of the work you've done. And so here I just flash up some of the titles from various manuscripts that we published based predominantly on that Illumina machine, the short read sequencer. And this is looking at viral genomes. This is reporting bacterial genomes from a wide range of environments and different projects that we were interested in. Okay, so what does the typical workflow look like if I'm doing one of these projects? So normally we'll start off with a sample. So here I've just got like a bunch of pictures of petri dishes with bacteria streaked out. But you would then cultivate your microbe, say in a liquid broth, little shake flask. You'll collect that biomass. And what you guys are going to do in the lab today is you're going to use one of these very friendly, commercially available kits to extract the DNA from a microbe. So there you end up with your DNA in a little tube. And from here, the workflow can deviate. So, for what we're talking about today, you might go on and sequence the DNA. But really now you can, say, clone the DNA like I described earlier, and you can go screen it for anything that you want. If we're sequencing, then as Shane described earlier, you'll have computational platforms where you want to put that genome back together, and then you want to analyze it in some way. You want to look at the diversity of the microorganisms in your sample, for example, or you want to compare specific gene sets to say how similar or different they are. So with the idea that you want to sequence these very long pieces of DNA in mind, if you're using the Oxford Nanopore technology, you also have to start with, you have to manage to isolate large pieces of DNA. Of course, in isolating the DNA, if it all shreds to pieces, you can't get it to go back together. So it's like Humpty Dumpty. You have to keep it intact from the start. So as I've described, today there are these commercial kits available, and here I just show you some agarose gel images. Any idea what an agarose gel is? Completely lost? No idea? Some idea? Nothing. So agarose gel is literally like jelly that you can buy at Spar, and you boil and you chuck it in there. So it's a sugar molecule. You dissolve it in hot water and you can cast it into a little plastic tray. So it just solidifies like a gel. You can then have these little holes at the top where you put your DNA sample in, and you can apply a voltage across this jelly. And because DNA is negatively charged, it'll start to migrate from the positive terminal to the negative terminal. And so it'll cruise through this gel matrix. Now DNA, as I showed you earlier, is this long, floppy strand. And so as it cruises through this gel matrix, the bigger pieces get retarded because they have difficulty moving through the matrix, and the smaller pieces run ahead. And in this way, we can actually separate the DNA based on its size. So yeah, just showing you some examples. This is beautifully intact DNA. If it was all degraded, you get like a smear down the lane, almost like what you see over here. So this is what you guys are gonna do. All right. So just to take then, shift gears again and go back to the antimicrobial discovery story. When you go about trying to find novel antibiotics, you would normally take one of two approaches. So this is the classic approach. This is how people did it from, say, the 1920s and 30s. It's a chemistry-based approach. So you would grow your bacterium, you would centrifuge it, get rid of all the bacterial biomass, and then in the culture supernatant, you would expect that there might be some antimicrobial compound produced by the organism. And you would then use your classic chemistry techniques, so you can do organic extract, so you can say add ethyl acetate or some other organic solvent, and the compound of interest might partition to that. And you can then use mass spectrometry and NMR to try and get down to a single compound that you then find is antimicrobial. But today, with the advent of next generation sequencing and bioinformatic approaches, we can also take a so-called bottom-up approach. So now if I sequence the genome of a bacterium or some other organism that produces these antimicrobial compounds, I can use bioinformatic tools to try and identify sets of genes that could be responsible for making the antimicrobial compound. I can go and whip out those genes, put them in my favorite bacterium, and I can try and express them there to then produce the compound. All right, so let me just show you how this sequencing business works. All right, so here is the baby sequencer from Oxford Nanopore. So this is called a flow cell, and right there, and maybe Anya's got one for us and we can show you guys. So here comes a piece of DNA, so that's the blue bit. I'm going to try and narrate this. So how nanopore sequencing works is you have a number of like proteins, so they call them pores, and your piece of DNA is actually going to zip through the pore. So there is a so-called motor protein that attaches to the end of the DNA, and as it unwinds the DNA, it actually forces the DNA through the pore. So the flow cell is engineered such that it'll measure the electric current as the DNA moves through the pore. So you see all these little squiggly bits. So those are meant to be the ions that pass from one side of the membrane to the other side. And as the DNA moves through, it'll disrupt that ionic current. And the flow cell is engineered to measure the changes in the electrical current that comes through. That electrical current is then converted to A, G, C, and T, the sequence of the DNA. So just to reiterate, so you've got a motor protein that sort of unwinds the DNA, moves through the pore, disrupts the ionic current, and the electrical signal gets converted to the base calls. This doesn't seem all that impressive, but let me impress on you that this is absolutely mind-blowing stuff. So just to fill it in a little bit further, the protein that they used initially is something called hemolysin. It has nothing to do with DNA. That protein was never evolved or engineered to interact with DNA in any way, shape, or form. They just picked it because the cross-section of the pore is big enough for a single strand of DNA to go through. The only reason we can convert the electrical signal to base calls is because of machine learning. They had to attach a motor protein to the DNA to make sure it goes through the pore slow enough that they could actually do this conversion. There is so much technology that went into the development of this. It's absolutely crazy. All right, so I'll just quickly show you some of the machines that they sell. So this one you will get to see in the main lab. At UWC, we've got the P2 Solo, one of these little guys, and we've got some of these, the Mark 1Cs. So this can take 2 of these Promethean flow cells, and each flow cell, they reckon, is good enough to sequence a human genome at 30 times coverage. So it's pretty impressive. And then these little guys are what normally we use to, if we have small cohorts of bacteria that we sequence, about 12 bacterial genomes per flow cell. We'll ask Anya if she's got some examples to show you. All right, so when we're looking for these antimicrobial genes, it is now established that they tend to come as these things called biosynthetic gene clusters. So if we look at a bacterial genome for genes that could potentially make some sort of antibiotic compound, then they come in these BGCs. So up top here, I've got what's like an idealized biosynthetic gene cluster. So normally we'll have core enzymes that are responsible for making the sort of main scaffold of the compound. We've got some biosynthetic tailoring enzymes. So once the core scaffold of the enzyme is made, maybe this enzyme is responsible for putting a methyl group somewhere on the molecule, a CH3, and that'll change its properties. We've got regulator enzymes. So this protein might come and bind to the piece of DNA over here, where you see the 2 genes read away from each other, to control the gene expression, because you may not want this pathway being expressed all the time. You've got a transporter gene. So if the compound that is made will kill the host, of course that's bad news. Now, I'm not going to live anymore. So if you have a transporter or even a self-resistance gene, then these 2 can work together to either pump the compound out of the cell or to inactivate it if it's inside the cell to keep the cell alive. So not all of the pathways have all of these features, but that's sort of a generalized, idealized version of it. All right, so then to Kalk Bay. So a couple of years ago, and thanks to Diplomix, who supported us, we had the idea to go and sample some of the sea sponges and sea urchins that you find down there, and try to get the associated bacteria out, cultivate them in the lab, and then look for these biosynthetic gene clusters in the genomes. So here we just got a video of us sampling. Yeah, this is my normal gear. This is what I look like every day. Made special effort for you guys. But yeah, so as you can see here, from this sponge, if you squeeze it, there's this brown soup that comes out of it. And if you go check that under a microscope, it is just teeming with life. So you get everything from the viruses to bacteria, micro-eukaryotes. And those are the guys we tend not to be interested in. We're all about the bacteria. So we collected a whole bunch of those and took them back to the lab. And so here's the workflow for that. So we've got the sponge, squeeze out the juice. We'll then plate that onto bacterial growth media. We'll leave it to incubate for a little bit. And of course, we start seeing some weird and wonderful colony shapes, morphologies. See this bullseye guy? There's a whole science and an art actually to cultivating these microbes on media. And part of the reason why metagenomics was developed as a tool is that we've come to realize that we can cultivate a very small portion of bacteria that you find in any environment in the lab. The vast majority, greater than 99%, we cannot culture, but we can get access to their DNA and we can use a surrogate host to try and express it and find these genes of interest. All right, so if we can cultivate it, we'll culture it up. We'll extract the DNA, that's what you guys are gonna do, sequence it, and then we use a piece of software called anti-smash to look for these biosynthetic gene clusters. So what I've got down the side here is just a table. I appreciate you guys probably can't see, but these are just some of the individual bacteria that we sequenced. And just to say that there is huge diversity in terms of bacteria. And let me find an example. So up here, so you guys know that for any plant or animal or microbe, we've got the binomial system of naming them, right? We've got a genus and a species. So over here, the genus JAHTBL01 species. We are finding new bacteria so fast with the sequencing technology. We can't keep up with naming them. There are one or two other examples as well. So the rate at which we gathering data is far outstrips our ability to analyze it in many respects. And that comes back to the points that were raised earlier. All right, so I've just given you some examples here of these biosynthetic gene clusters that we found in some of these bacteria. And when we compare it to things that are already on huge databases, the things that we found here in Kalk Bay are really new and novel. So, and if you start to think about it, some of the talks that you saw, South Africa's got a huge coastline. Here, we drove down to Kalk Bay and we took one sea sponge and we cultivated some of the microbes out of there. If you start extrapolating that to the whole of the South African coastline, there is a huge amount of microbial diversity, and heaven only knows what else we'll find in there. So the pie chart here just shows the sort of breakdown of the different classes of compounds that we found. And so this big light blue slice are the polyketide synthases, non-ribosomal peptide synthase hybrids. So non-ribosomal peptides and polyketides, these are two classes of compounds that are well known to be biologically active. So that's not just antimicrobial. It can have anti-cancer properties, it can be anti-inflammatory. There's a whole host of benefits that you could derive from these type of compounds. Right, so we can also compare the different suites of genes to each other in something called a network analysis. So this is a pretty basic one that I did when we started this project. And so essentially every dot represents one set of these genes that you find in the organism. And then of course the lines show you whether there's some sort of relationship between them, whether they are highly or more distantly related. And what you can see is that we've got a lot of dots that aren't connected to anything. And that's great because it means they're new and novel and they're not necessarily related to anything that we've seen before. So here I put some sort of annotation. These are pathways that are present in the MyBig database. So this is a database of suites of genes that show some sort of biological activity. And again, you can see that at least for the genomes that we represented here, there aren't that many that are related to something already on the database. And then here is a slightly updated one. I appreciate you guys can't see all the dots. And this is, so as we go along and we sequence all these bacterial genomes, I tend to do this analysis again and again and again. So this was also like quite some time back. So at some point in the future, those 500 genomes that we mentioned, I'll put through this pipeline again and it'll keep growing. Another type of analysis we can do on these genomes is we can compare the whole genome. And so here, the example that I show is if we compare the genomes and ask, are these bacteria similar at a species level? So that is what is represented over here. Then essentially, this is the most modern tool for classifying bacteria. And it says that all these bacteria, these 3 bacteria, are the same species. But if you physically compare, if you line up the whole genome, you can see that there are vast stretches of the genome that don't correspond to each other. So this is a so-called pangenome. This would be the core genome, the part that's conserved between all 3, and then you've got these areas that are extra genes. And so even though at a species level they're all the same, functionally they can be very different. And very often we find that the biosynthetic gene clusters will make up the remaining part of what is different between them. All right, so that was in the case where we could cultivate these organisms on an agar plate. And so next, I'll just tell you about the case where if we can't cultivate them, what happens if we just squeeze out that juice, extract all the DNA, and we apply this metagenomics tool, what do we get to see? So this is just a depiction of that whole process. So you've got this mixture of microorganisms. You can extract the total DNA from there. You can prepare a sequencing library. Here we've got our nanopore, and you can sequence all of the DNA in the mix. You can then try to reassemble everything. And today we've got software tools that'll actually allow us to pull all the microbial genomes apart from this mixture. And then of course we can classify and annotate the different genes and genomes, and we can look at things like relative abundance. But of course we can also go hunting for our antimicrobial suites of genes. All right, so when it comes to metagenome extraction, more often than not, we tend to use classic techniques. There isn't always a commercially available kit for this. But in this case, people have developed such a kit, and so we use that on this occasion. So here we are earlier this year, back in Kalk Bay, sampling that same sea sponge. And so now, instead of trying to cultivate the organism, all we did was to filter out all the organisms that we don't want, these micro-eukaryotes. And from there, I used the kit, and we managed to extract DNA of sufficient quality to then go on and sequence all of that. And so if we look at the data that we got from there, now we can start to have a look at what is the diversity of the bacteria represented in that mix. And something that you guys won't necessarily appreciate, but when you do these big data projects, it all is dependent on having really well curated databases that you can compare your data to. And so I made special effort on this occasion. There's a very large database you can download. It takes forever. But the result of that is that 99% of the reads in this data set we managed to classify, which is really, really good for metagenome. And so what we do get to see is that we've got a bunch of Vibrio species and Pseudo-Ultramonas. I appreciate this means nothing to you, but these were some of the bugs that we could see in culturing as well. What was really surprising is this guy that seems to make up a really big portion of the data. This organism, Fusobacterium ZRK30, is an obligate anaerobe. It cannot grow in the presence of oxygen. And that is really surprising to find in a sea sponge. So sea sponges are thought of as things that are very oxygenated. They will filter a lot of water through to get the nutrients that they want. And so it is not appreciated that you've got pockets inside these organisms where you've got anaerobic environments, and certainly not to that degree. So yeah, that was really interesting for us to find. And we would never have seen that guy in culture because you have to culture it anaerobically. So if we try to reconstruct some of these bacterial genomes, again, it's a lot of, what did you say? It's word vomit or whatever. But yeah, so we managed to reconstruct very many of the genomes that we find in there. And of course that bodes well if you go hunting for these gene assemblages. So one of the classes of compounds that we're really interested in are things called lantipeptides. So I guess you guys know what a protein is, right? And a peptide is just a short version of a protein. So what these lantipeptides look like is they are, say, 6,000 Dalton long peptides, and they're composed of a leader and a core. And it's normally the core peptide that has some sort of biological function, whether that is cell-to-cell signaling or is antimicrobial or what have you. So in the case of lantipeptides, they are co-expressed with post-processing enzymes, and they, depending on the post-processing enzyme, they come in different classes. So what these post-processing enzymes do is they install little bonds, chemical bonds, called lanthionine bridges. So here you've got a more detailed view of that. We don't need to worry about the details too much. But that's followed by the cleavage separating the core peptide from the leader. And it can then go on and do its particular function. So as part of a EU-funded program called Blue Remediomics, and as part of why we're here today, we identified several of these lantipeptides from those microbes that we isolated from Kalk Bay. And currently in the lab, we're at various stages of making gene constructs, expressing them, purifying them, and then testing them for all sorts of bioactivity. And then really interesting, from the metagenome, so this is where we just squeezed out all the juice and sequenced everything, we managed to identify pathways that we didn't see from the cultivated genomes. So here I just show you one example. So this is a type 3 lantipeptide, and this is the, what the chemical structure of the peptide should look like when these lanthionine, or in fact, it's not lanthionine rings, now it's a labionin ring. So this is a specialized version of a lantipeptide. So yeah, these constructs are on their way from America. Once we get them, we'll pop them in our favorite bacterium, express them, and see if they have some sort of biological activity. All right, then I'm gonna leave it there for now. I'm not sure how I'm doing for time, but just to say a big thank you to Shane and Diplomix. They've supported us for a very long time with all of the gear necessary to sequence all these genomes. And yeah, just thank you very much for your time as well.